Hello everyone, in this video I'm going to be talking about the structure of mitochondria. Now I'm not going to go into detail about many of the functions of the mitochondria because that would be a whole video in itself. Instead I'm just going to outline the basic structure and what can be seen from this image obtained from an electron microscope. So this electron micrograph picture that we've got here is of a mitochondria, and I've labelled 5 particular parts, A, B, C, D and E. And I'm just going to explain which bit is what, essentially. So let's start with A. A is what we call the intermembrane space. So this intermembrane space is what our part A is. The intermembrane space, it says there, allows for hydrogen ions or protons to be pumped into that space to set up concentration gradients for what is known as oxidative phosphorylation. Now, oxidative phosphorylation is a stage in respiration. Now, mitochondria is the site of aerobic respiration, and oxidative phosphorylation is one of the key stages in aerobic respiration. So that's just an intro as to what that sort of region is. So on this electron micrograph, A is known as the intermembrane space. And as part of the process of respiration, hydrogen ions are going to be pumped into that space. Ultimately, I'm not going into detail, but ultimately all those hydrogen ions will then rush back from that space to where they came from, activating a particular enzyme called ATP Synthase, which will generate ATP for us. So ATP is our source of energy, if you like. So respiration, when we say, is a site of energy production, these hydrogen ions being pumped are key in that. B on this picture, B represents what is known as the matrix, which is fluid inside the mitochondria, and it's full of enzymes needed to catalyze the link reaction and Krebs cycle. Now, the link reaction is one in respiration where pyruvate gets converted into something called acetyl-CoA. And this whole process is known as oxidative decarboxylation. Now, that link reaction is almost like a precursor to what's known as Krebs Cycle. Krebs Cycle is the big cycle in respiration where we use that acetyl-CoA and we get ultimately the production of ATP. So, like I said, I'm going to, in a separate video, talk about Krebs Cycle, but that's what those enzymes are used for. They're used in those respiration processes known as the link reaction and in Krebs cycle, and they're found within that matrix, that fluid. Let's think about the cristae. Now, the cristae is what I've labelled here as C. The cristae are little projections of the inner mitochondrial membrane. Because if we think, now in textbooks on the internet, when we sometimes, even on my videos, I've sketched the mitochondria a bit like this. It's a very simplistic drawing of the mitochondria. And what we've got is the matrix inside here. And notice I've drawn 2 black lines. I've drawn this one, which is the outer membrane, and then I've drawn this black line here, which is an inner membrane. That, if you like, is our cristae. They are projections of this inner mitochondrial membrane, and they are designed, as we've put here, to optimize the surface area for oxidative phosphorylation. So that phase in respiration. So as part of respiration, to maximize the process and energy production, we have these projections of that membrane called cristae. Like I said, this is just to outline the basic structure of the mitochondria. So we've put in place an intermembrane space, the cristae, so projections of that inner mitochondrial membrane, and the matrix, the fluid found within it. Then there are just 2 things I'd like to just comment on. Where I've put D, that is to represent DNA, because there is mitochondrial DNA here. We have naked loops of DNA found in the matrix. And E, not amazingly clear, but where I've got E, that is where we have what are called 70S ribosomes. It's a smaller type of ribosome, and that is the site of protein synthesis. So that's the basic structure of mitochondria. Two questions that students often ask me, in fact, when we look at the structure of mitochondria, is how can you extract them from a tissue sample? Like if I wanted to look at and get this particular image, for example, how can I extract mitochondria from all the other organelles? And it's via a process known as cell ultracentrifugation. So it's called ultracentrifugation. And that's a process, ultracentrifugation is a process where you actually break open the cell, separating out all of the organelles, and you spin the actual sample that you have in test tubes in a machine known as a centrifuge, and you separate organelles out by their size and weight. I have a whole separate video on centrifugation process, so I won't delve into too much on this video. But using that process, you can actually separate out mitochondria from things like the nucleus, the endoplasmic reticula, the Golgi body, lysosomes, ribosomes. And another question that students tend to ask is, based on the fact that ribosomes in the cytoplasm of eukaryotes, so typical animal plant cells, ribosomes tend to be of the ATS type. But when you look at the ribosomes in the matrix of the mitochondria, they're of the 70S type. Now these 70S ribosomes typically we see in prokaryotes, like bacteria, for example. So an interesting question arises, why would ribosomes in cytoplasm have ATS and yet those in the matrix be of 70S? And there's actually a theory that's been put forward to suggest that mitochondria were early primitive prokaryotic cells, almost like mini bacterial cells, if you like, that got enveloped into larger eukaryotic cells and remained. So it's an interesting point to just note that the ribosomes are of that different kind of type. They're of the smaller 70S type. So it's just a quick video on the structure of mitochondria. And in another video I'll be talking more in detail about the functions of mitochondria, namely aerobic respiration and maybe something called apoptosis, programmed cell death we'll look at, because it's also involved in cellular metabolism and regulating that, and natural steroid synthesis. So the mitochondria have so many different roles we'll look at in another separate video. Okay, I hope all that helps.